Hello, and welcome back to Introduction to Genetics and Evolution. We're at a transition point in the class now. We've already gone over a lot of basic transmission genetics. We've talked about things like recombination rate, we've talked about genetic mapping, things like that. What you'll start to see now over the next couple of videos is we'll still be talking about topics related to genetics, but we'll gradually be getting significantly more evolutionary, and we'll talk about some things having to do with evolution. Starting with today's talk. Today we'll be looking at the different contributions from genetics versus the environment to various traits. Basically, how do we tell if each of the two contribute to a particular trait? How do we tell that some trait has a genetic contribution versus an environmental one? Well, the media often portrays this genetics versus environment, or nature versus nurture, as they like to say it, as some sort of raging debate. And in fact, I've had people on airplanes ask me, like, where do you stand in this debate? There is no stance. That's just ridiculous. It's nearly always somewhat of both. But we do want to be able to actually identify that there is a genetic component or an environmental component, because there are a small number of traits that only have one. Let me start with this example. This is a picture from my backyard. These are some corn plants that I was growing right next to our deck there, as you can see. Now, what I did here to make it a little bit easier to see is I put these white bars, as you can see here, just depicting the size of the corn plant next to it. So you see I have some that are about this tall, some that are about this tall, and right next to the deck I have some significantly shorter ones. So why is that? Why do you think there is this difference? Then it's not just one corn plant. There's a bunch of them that are all about the same height. A bunch of them, they're all about all the same height. A bunch of them, they're all about the same height. Do you think I split up the seeds and put all the tall corn over here, the medium corn over here, and the short corn over here? Probably not. Do you think I watered them differently? No, they're all pretty much watered the same. Well, if you think about it for a second, the answer is kind of obvious. That these corn plants out here were getting a lot more sunshine, whereas these corn plants were getting a lot less, because you can imagine as the sun was going over, there's still, all of them are still getting sun. When it gets over way over here, these ones are still getting sun, but the ones up against the deck are not getting any sun. So there clearly is some sort of environmental component to this corn. But how do we quantify this? And more fundamentally, why do people even care? Why do people want to quantify whether there's a genetic versus environmental component? Well, there's a lot of reasons. As you'll see later in the class, you'll see that we want to be able to predict evolutionary change, right? And evolutionary change requires that there's some sort of genetic component. You can't have natural selection or evolution by natural selection unless there is some hereditary basis to the traits that are being selected. Okay? This is often also used in animal and crop breeding. So let's say, for example, you're a farmer and you want to breed for corn that grows tall. And let's say that there is basically no genetic component to it. Do you want to spend a lot of energy trying to select when there's in fact going to be no response at all? Or let's say there's no environmental component. Do you want to spend a lot more resources for excellent fertilizer and things like that when it's not going to matter? They're all going to grow to the same height anyway. No, that basically you want to know this sort of information so you can spend the least amount of effort or energy possible for the best yield. This is true for both animal breeding and crop breeding. You want to know if the environment matters, and you want to know if it helps to pick parents that have particular traits. This is also very important in the context of medicine. Let's say, for example, you're a doctor and somebody comes to you and says, if my mom and dad had peptic ulcers, am I likely to get them? Basically, is there some sort of genetic predisposition to peptic ulcers? You want to be able to have an answer to these things, and this is where this kind of work is very useful. So let me emphasize again, this is not a pure dichotomy. This is very rarely an either-or question, but is usually some of both. However, one may contribute way more than the other. So something like, for example, eye color. Yes, there's probably some small environmental component to that, but there's a very strong genetic component to eye color. That, if I could lie in the sun staring at the sun forever and my eyes would not turn blue. Something like that. Similarly, HIV status. There are some genotypes which are slightly predisposed to being less likely to getting infected with HIV. But there's obviously a huge environmental component that your behavior is very likely to affect how likely you are to get HIV. So let's dive into this. First, do genetics contribute? The way people usually identify some sort of genetic contribution, and I mentioned this in the beginning of the genetics section, is that there's some sort of parent-offspring resemblance, right? That this could indicate that genetics are important because people who are related tend to look more similar to each other than to just somebody at random from the population. However, this is not a perfect measure. And part of the reason for that, and this is something we'll come back to a little bit later on in this set of videos, not in this video in particular, is that parents and offspring often share part of their environment too. So how do we separate these two things? How do we distinguish? Well, I'm going to show you a couple of different means. In the first part here, I'll talk about resemblance between relatives, and I'll use examples of identical versus fraternal twins, or monozygotic versus dizygotic twins as an example. And I'll use that just as an illustration, and then we'll come to the more meaty parts, which is common garden experiments or reciprocal transplant experiments. And those use the same principles as the sort of twin examples. And you'll see this in just a moment. The idea here is we want to separate genetics from environment. We want to keep one side constant and vary the other and see if there's an effect. If we're keeping the environment constant, we vary the genetics and we see an effect, then there must be some genetic contribution. If we keep the genetics constant, we vary the environment and we see some sort of effect on the phenotype, then there must be an environmental component. That's the basic principle. So looking at the two types of twins I mentioned, monozygotic, which are identical. These are ones that came from a single sperm and egg that then separated out and they shared a single placenta. These are genetically exactly the same. Okay? Dizygotic or fraternal twins are actually genetically no different from any other brother or sister. They're separate sperm and egg fertilized. You can never have identical twins that were different sexes because you can't have different sexes that have exactly the same genotype. Famous example of some twins are the Olsen twins here. They, by the way, are fraternal rather than identical, just so you know. Now, we can leverage this difference between the monozygotic and dizygotic twins. We predict if there's a genetic component to some trait. So let's say you're looking at height. Let's say there's a genetic component to the trait. The monozygotic twins should be more similar to each other in that trait, such as height, than dizygotic twins. On average, if you look across monozygotic twins, they should be very, very similar. Dizygotic might be a little bit more different because the dizygotic twins don't have exactly the same genotype. They have some different genes, whereas the monozygotic have exactly the same gene. And we're assuming these twins are basically reared in the same environment or close enough. There's no particular difference. Like if you're having kids, if you have monozygotic twins versus dizygotic twins, you probably don't treat them any differently. You treat them the same because they're your kids. So the way we measure this, the way we measure how similar or how different you are is using correlations. So using correlations and traits is that we do this. And this gives you some measure of how well traits match between groups. I know some of you may have had a statistics background, some of you may not. So for those of you who already know some statistics, I apologize for this review. Correlations are given a numeric score, which is often labeled an R. And that R ranges from -1 to 0 to +1. Now what this does is indicates how closely one predicts the other. So let's say, for example, you have a perfect positive match. That would be an R of 1. So an example of this would be your height in centimeters to your height in inches. One perfectly predicts the other, right? That you have this absolutely perfectly linear correlation between the two. Okay. In contrast, let's look at one that's not quite so strong. Here's one. Let's say you compare your height to your brother's height. You may see something more like an R of 0.7, where on average, if you have a tall brother, you're likely to be tall. On average, if you have a short brother, you're likely to be short. But it's not this perfect correlation. There are some individuals who have a tall brother but are a little bit on the short side and things like that. So it's not perfectly predictive. And again, in this case, some of this may be from genetics and some of this may be from shared environment. That's why you do see some sort of association here. Now let's go a little bit further. Let's imagine there's no correlation. You may see this in the context of your height to your neighbor's height, your next door neighbor's height. It's quite possible that you'll have some very short neighbors, you may have some very tall neighbors, but there's no reason to expect that their height is any more or less like your height. So this would be a case where the correlation is 0. Now you may be wondering, well, I've only done this range from 0 to 1. What about the negative part? Well, negative is when you have literally a negative prediction, such as daily high temperature to morning rainfall in the month of April. That you may have a lot of rainfall associated with low temperature, but as rainfall goes down, you get a higher and higher temperature. Okay, so this would be an example of a negative correlation, or a -0.5 in this particular example. So if that gives you an illustration of what I mean when I say correlations. So let's come back and use this. The prediction, as I said before, is that if there's a genetic component to a trait, monozygotic twins should have a stronger correlation to that trait than dizygotic twins. Right? The monozygotic are the ones that are genetically identical, right? And the dizygotic ones have some different genes. And we're assuming that the environment is about the same in both. So what do we see with the data? Well, if we look at correlations with twins, we often see this difference between monozygotic twins and dizygotic twins. Here are a couple of examples. One is for IQ, or the supposed intelligence coefficient, that monozygotic twins tend to score very similarly on these tests. The correlation is 0.85, whereas dizygotic twins have a correlation of 0.42. This suggests there is a genetic component to what is measured here as IQ. Same sort of thing for some diseases. When we look at gastroesophageal reflux disease, monozygotic twins have a 0.29, dizygotic twins have a 0.13. Again, we see this higher association when they're genetically the same than when they're genetically similar but not identical, which means that there is some sort of genetic component. There may be an environmental component. I should stress this. None of this proved the absence of an environmental component. All it shows is the presence of a genetic component. I should stress that. Okay? Now, since this didn't prove the absence of an environmental component, how do we look at that? Well, what we can do there is keep the genetic relations constant but vary the environment. What you do is you look at monozygotic twins reared together versus monozygotic twins that were reared apart. And your prediction is that monozygotic twins reared together should have a higher correlation in these traits than monozygotic twins reared apart if there's an environmental component. The reason is those monozygotic twins reared together have exactly the same genes and the same environment, or very, very, very similar environment. Whereas monozygotic twins reared apart have exactly the same genes, but somewhat of a different environment. Now, this has been done quite a bit, especially in the context of psychology, but in other areas as well. So here are some results. Looking at the body mass index, or BMI, monozygotic twins reared together have a correlation of 0.74. In contrast, monozygotic twins reared apart have a correlation of 0.70. This suggests there may be some environmental component, but it is surprisingly very small. Very small, surprisingly small environmental component. You may think this is unexpected for something like body mass index, but in fact, for this particular study, that's what they found. In contrast, for verbal ability, monozygotic twins reared together had a correlation of 0.76, whereas monozygotic twins reared apart had 0.51. So this suggests a very strong environmental component to verbal ability. Now we can again do the same thing with the monozygotic versus dizygotic. So looking at this showed a strong environmental component. This also shows a strong genetic component because again, the monozygotic together versus dizygotic together means the environment's the same. Mono versus di is changing the genetics. In this case, we see a pretty large genetic component and a moderately large environmental component for the same trait. Again, seeing this difference doesn't necessarily mean that it doesn't mean the absence of a genetics component. It just means the presence of an environmental one. Okay? So again, what we try to do is we try to control for environment and vary genetics and see if there's an effect, or control for genetics and vary the environment and see if there's an effect. Well, the same principles are done when you're looking at animals or plants and things like that, and not involving mono and dizygotic twins. And in fact, these have been done for quite some time. Let me show you some classic work. This is from the 1930s, work done by Clausen, Keck, and Heisey. And they were interested in these differences among plant forms identified from different elevations. So they got some plant forms from, as you can see here on this elevational gradient, from timberline, which is very high in the Sierra Nevada of California, from Mather, which is moderately high in elevation, and from Stanford, which has a very low elevation, just above sea level. And what they found is that the plants, these are the same species, looked very different in these different forms. At the high elevation, they tended to be kind of thin, sparse, not particularly bushy. Here's a color photograph of the high elevation form. As you got to lower elevations, you got this much bigger, bushier form down there. Is it genetics or is it environment? Well, they pioneered a set of work that was extraordinarily influential. So one thing they tried were these common garden experiments. These had actually been done by other researchers in the past, but they applied this to these particular plants in California. Essentially what they do for this is they grow plants obtained from different places in a single environment. So the idea here is they're trying to keep the environment constant, but getting these plants from different places, these likely have different genes, right? Because they're coming from different places. They likely have a different genetic makeup. So if the form difference between, say, the high elevation plants and the low elevation plants was all environmental, right? If you grow them in the same place, they should all look the same. In contrast, if this form difference between the tall and short plants was all genetic, then the plants should look quite different. Even if it's partially genetic, they should look quite different. This is very similar to the concept of mono versus dizygotic twin studies because, again, we're using unrelated plants, so they have different genes, but looking at them in a constant environment. Well, what did they see? In fact, they found there was a form difference apparent even when grown in the same environment. So these are ones from the species Potentilla gracilis, and grown in one spot in Mather. So this is that intermediate elevation spot. They found quite a big difference in form. And this suggests this form difference does have a genetic component. It does not prove there was no environmental component. It only proves there is a genetic component. Okay. Well, how do we do the other half of this? Well, the other thing you can try is what's referred to as a reciprocal transplant experiment. In this case, you grow plant forms obtained from a single environment. So since they're from the same place, we're assuming the genetics is fairly constant. And you grow them in several different environments. So you're taking all these ones that have the same form, and you're growing them in different environments. So maybe you take it from Mather, grow it in Timberline, in Mather, and in Stanford, for example. If part of the form difference is environmental, then they should in fact look different. In fact, if the form difference was purely genetic, then all the plants would still look exactly the same, right? Because they're all derived from that same place. Okay? And this is the same concept as these twins reared together versus apart, that we're using the same genetic makeup, right? And we're varying the environment in which the plants are grown. And in fact, with this, they did still see an effect. The form difference for the same type grown in different environments was still apparent. This is showing Potentilla glandulosa. And this was grown at Stanford, Mather, and Timberline. And you note again, the Timberline ones are quite small. The Stanford ones are much bigger and bushier. This suggests this form difference does have an environmental component. Now, everything I've shown you so far is how to tell, is there a genetic component? Yes or no. Is there an environmental component? Yes or no. And remember, just because you say yes to one does not mean you're saying no to the other. Because quite often, as you saw with these examples, there is some of both. Now, what you probably want to do is to quantify how much of the variation that you see is genetic versus environmental. Well, that we'll come to in the next video. Welcome back to Introduction to Genetics and Evolution. In the previous video, we were looking at this dichotomy between genetics and environment. And what we did in that context was trying to figure out whether or not there's a genetic component to particular trait differences you see, or whether there's an environmental component to trait differences we see. Now, what we want to really do is to actually get this part. We want to say how much do each contribute to particular traits of interest. We want to quantify how much they contribute. Now again, when we're inferring whether genetics or environments contribute, typically the answer is very often both are contributing. In that regard, it's not really that satisfying to just say, yes, there's a genetic component, or yes, there's an environmental component. It's much more satisfying to actually infer their relative contributions. To say for a particular trait, this is something that 90% of the variation you see is genetic, or 90% of the variation you see is environmental. Well, this comes to the concept that we'll refer to as heritability. I'll define that in just a moment, but first, let me go through a couple of quick mathematical concepts that will be relevant to this lecture. Let's talk about mean and variance, please. And I apologize for those of you who already have a lot of statistics background, but I just need to go over this. Now, most traits you look at tend to be variable, that not everybody has exactly the same trait value. So you can use height as an example there. Now, if a trait is continuously variable, right, meaning that you have all these different steps, so something like height, you can be 5 foot 9 inches tall, 5 foot 10 inches tall, 5 foot 11 inches tall, 5 foot 11 and a half inches tall. Then we can calculate a mean and we can calculate a variance. Now, many of you are familiar with variance in the context of just thinking of it as the spread of values. And I can depict this. So let's say you're looking at height in the classroom. So let's say this is the height in the classroom. This is saying 5'0" to 6'2". This is 5'0" to 6'2". What is the mean? Well, in both of these classrooms, it looks like the mean is right below 5'7". Okay. In contrast, which one has more variance? And again, if we think of variance just as spread, we see here the graph on the left has a lot more variance in height than the one here on the right, where basically everybody is 5'7". Well, making this more quantitative, the mean is simply the numerical average. That if you have three numbers, 1, 2, and 3, the average is 2. You sum them all together and you divide by the total number of values. The variance is a spread, or basically how spread out individual measures are from the mean. The calculation for it is this: the summation of all the individual measures minus the mean, and for each one of those squared, divided by the total number of measures. Okay, that's the formal formula for the variance. So if you had 10 individuals, all with height 69 inches, okay, so let's imagine that's their height. Everybody has a height 69 inches, and there's 10 of them. What is the mean going to be? Well, the mean's obviously going to be 69 inches. What is the variance? Well, in fact, in this case, there's absolutely no spread whatsoever, because we said everybody has exactly the same height. So the variance would actually be 0 in that example. Let me give you a few others. Here are two sets of measures. Let's say these are, again, heights of people in a classroom and height in inches. And I have the formula here in case you want to refer back to it. So in classroom 1, 63, 65, 67, 67, 69, 71. Those are the heights you see. For classroom number 2, 65, 66, 67, 67, 68, 69. Which one has greater variance? Is it classroom number 1, classroom number 2? Or are they equal? Well, you can probably do this without the math. Let me encourage you to try that without the math. Answer on the next question within the video here, and then I'll show you how to work it out both with the math and just eyeballing. Okay, well, I think eyeballing this is a pretty easy problem. Looking at the spread here, they're both perfectly symmetric, right? But this one goes further out. This one is much more clumped together. So the answer is number 1 definitely has more variance than number 2. Well, let's try this out numerically. So if we get the height in inches, the means for both are 67. That's just adding all the numbers together and dividing by the number of measures. The variance in this case comes out to 6.66 for classroom number 1. This is taking each individual measure minus the mean and then squaring it, adding all those together and then dividing by the total. So this is 6. This comes to 6.66. For those who are interested in sampling variance, no, I didn't put that in there. For classroom number 2, it comes out to 1.66. So clearly there's more variance in classroom number 1 than classroom number 2. Well, variance, and don't worry about the math for this right now. We'll come back to the math later. Variance, think of it, again, just as the spread that you see. As the variance is larger, there's more spread. As there's less spread, the variance is smaller. Okay? So what causes this variance in traits like height? Well, let's use this example right here. Let's say here on the y-axis, we're looking at number of offspring that have a particular height. And on the x-axis, we're looking at height, such that this would be short at this end, and this would be tall at this end. Okay? So we see here, in this particular case, there's some variance in the phenotype being height. That we have some individuals that are on the tall end, some individuals that are intermediate, some individuals that are short. Now with this example, we're looking at 2 alleles at a single gene that are controlling height. We're assuming there's no dominance to it, and there's no effect of the environment. So that's why these are perfect lines, that if you're little a, then you're exactly that height always. If you're big A, little a, there's more of you, and you're exactly that height. If you're big A, then you're exactly that height. So in this case, there's no effect of the environment, and there's some variance there among the individuals. That variance in this case is all genetic, right? Because we said there's no effect of the environment, so all the variance is genetic. Now what if we saw this? This is still 2 alleles at a single gene controlling height, and there's still no effect of environment, but we see a bigger difference in height between some of the individuals. In this case, the lower graph here has more genetic variance than the upper graph. Both of them still have no environmental variance, but the lower one has more genetic variance because you see there's this bigger difference in phenotype among individuals with no environmental part, and it's purely controlled genetically. It's controlled by your genotype at the A gene. Now what would happen if we said, well, there's a little bit of environmental variance here, and there's a little bit of mush? Well, we might see something more like this. In this case, big A individuals are still intermediate, little a are on average taller, and big A are on average taller. But we have some effect of the environment in here too, where some big A individuals might be almost as big as a big A individual. Some little a individuals might be as short as a big A. But what we've done here is we've added some environmental variance, some effect of the environment. There's still a genetic variance here because there's still a difference among the genotypes on average, but there's also some environmental variance. This is probably the more typical scenario where you have this mix of genetics and environment. Now, the formula that we use for this is very simple. VP, which is the phenotypic variance, so you calculate the variance in height, just like we did before in the beginning of this video. And we say that's the sum of that fraction of the variance that's genetic with that fraction of the variance that's environmental. It's a very simplistic formula that the overall phenotypic variance has a genetic component and it has an environmental component. Okay? Now, how do we calculate this? Well, that's the big question. I'm going to show you in this video one mean, and that's using an F2 cross between some strains that are isogenic. In the next several videos, you'll actually see other means for calculating heritability, and specifically what fraction is genetic versus phenotypic. Now, let's say that you're looking at genetic variation in the F2 of a cross, and let's say there's 6 genes for height. Let's start with the very artificial scenario that we know these 6 genes are involved, and you have capital A, capital B, capital C, capital C. It's just like the example we used a while back. So these are some 6-foot tall people, and this is the exact genotype that's causing that height. And let's say they have kids with these ones that are 5-foot tall. Okay? How much genetic variance is there in the tall parents? Well, we're saying there's none because we have their actual genotype there, and they're all exactly the same. In this case right here, we're saying there's absolutely no genetic variance in the short parents either. Right? What about the offspring? They're all heterozygous. Is there any genetic variance? The answer is actually no, because every one of the offspring has exactly the same genotype. Even though they're heterozygous, even though they have 2 different alleles, there's still no genetic variance in these F1s. In the F2, however, you'll have a lot of variance, some of which will be genetic, because you'll start to get some individuals that are big A, some individuals that are little a, etc. And if you're unlinked, then there's many, many possibilities. And you saw this before with the slide I used earlier. So let's do this cross. Again, we're saying there's no genetic variance in the parentals, in this parental, in the tall parents, or in the short parents. Between the parents, yes, sure, there's genetic variance, but we're not looking at that. We're looking within this parent, or within the F1s. There's no genetic variance. When we see this genetic variance in the F2, right, we see that there's this massive increase in genetic variance in here. We can use that. We can leverage it. Let me show you how. Let's say, for example, in actual height, among the tall people, among the 6-foot tall people, there is no phenotypic variance. This is important. I'm saying phenotypic variance. No phenotypic variance there. Remember, phenotypic variance comes from genetic variance and environmental variance. Since we already knew there was no genetic variance there, if there's no phenotypic variance, then there can be no environmental variance. Okay? So VP is 0, VG is 0, and VE is 0. This is true for all 3 of those. Now let's add another step here. Let's say there is a little bit of environmental variance. That's okay. I mean, we can say that maybe all these people aren't exactly 6'0", all these people aren't exactly 5'0", but let's say we know they have this particular set of genotypes. In this case, there's still no genetic variance, but there is some environmental variance. You note the environmental variance should be about the same for all 3 of these. Okay? We're assuming that these are all grown in, say, the same garden, or if they're people, they're obviously not grown in a garden, but you know what I mean. Now, what will happen is in the F2, even though you had a little bit of environmental variance here, but no genetic variance, in the F2 you'll have genetic variance and environmental variance. Well, we can leverage this, because up here, the phenotypic variance, which this you can measure, you can measure the phenotypic variance in the F1s or in the parents, right? You can measure just the way I was showing you with height, where it's the difference between individual measures minus the mean squared divided by the total number assayed. You can measure the phenotypic variance. In this case, you're getting an estimate of VE. Over here, you can again measure the phenotypic variance, and you're getting VG plus VE. So all you have to do is subtract this phenotypic variance minus that phenotypic variance, and you get VG, right? If you take this phenotypic variance, which is VG plus VE, subtract from it this phenotypic variance, which is just VE, you should get VG, right? Very simple. Well, again, these are the components we have, and we want to know how much is genetic versus environmental. How much is that contributing to the overall phenotypic variance? Well, the fraction of the total phenotypic variance that is genetic is called heritability. So heritability is called VG over VP, or you can think of it as VG over VG plus VE, because that's just synonymous with VP. So this ranges from 0, when there's absolutely no genetic component, to 1 when there's all genetic component. So you can say something, for example, is 90% of the variance is genetic, or 10% of the variance is genetic, using this very simple formula. So let's try this out. Here are some examples. Let's say you manually calculate the variance in the F1s to be 5. So here, this number is equal to 5. Okay? You manually calculate the phenotypic variance here in the F2s, and you calculate that to be 25. Okay? So what is the heritability? Well, I have the formula right here. Heritability is VG over VG plus VE. Well, we know VE, right? VE is 5. And we're assuming the same VE here as over here. We're assuming it's the same. So we're assuming the environment is basically the same in the F1 as it was in the F2, right? So the genetic variance must be what? Well, VE was 5. VG must be 20. So the heritability in this case, heritability is often abbreviated H squared, is equal to VG, which is 20, over VP, and we'll use this one right here, because we're measuring this in the context of the F2 is 25. So the answer in this case would be 80%, or 0.8. Now this may seem like an artificial scenario because what's happening here is you're starting with lines that you know have no genetic variance. Now in terms of human height, sure, you'd never apply this. However, this is something that is quite commonly used in the context of, say, crops, where you have this pure breeding tall corn crossed to pure breeding short corn. You want to look at the heritability of corn height or something along those lines. The same with model organisms like Drosophila, fruit flies, things like that. So this is used quite a bit, but it's not used in the context of, say, human height or anything dealing with humans. Well, how do we deal with things like humans? Well, that's the subject of the next video. Thank you. Hello, and welcome back to Introduction to Genetics and Evolution. Now, what we've been trying to do in the last couple of videos is quantify, when you're looking at variation in some traits, such as human height, how much of the variation you see is genetic and how much of the variation you see is environmental. Now, in the previous video, we estimated heritability, or that fraction of the overall phenotypic variation that is genetic, using an F2 cross. Here's the formula, by the way, for heritability. It's, again, that fraction of the overall phenotypic variation that is genetic. Okay? We estimate using an F2 cross. And we started off with tall parents that are all genetically identical to each other. So the genetic variance here is equal to 0. We cross them to short parents that are all genetically identical to each other. So within this box, genetic variance is 0. We got intermediates, the offspring here, from them. The genetic variation within these is also 0. They may be heterozygous for a lot of loci, but they're all heterozygous for the same loci in exactly the same way. Okay, so this is the F1s. From these, we got the F2s. In the F2s, we see quite a bit more spread than we saw in the F1s or in the parents. The reason we see more spread is because we've added this. Instead of VP just being VE, VP here is VE plus VG. We've added genetic variance here. From this, by comparing, assuming that this VE is the same as this VE, comparing the two VGs, we can get an estimate of heritability, as you saw. This is not very useful in the context of something like, for example, human height, because we don't have a bunch of people who are all genetically identical that are tall, and we can cross to a bunch of people that are all genetically identical that are short, and look at the offspring and then their grandkids. That obviously is not feasible. Instead, another way to estimate heritability is to use what we refer to as parent-offspring correlation. If we assume that all variation is genetic, and let's assume there's no dominance in this case, then any individual should have exact average phenotype of the two parents. So here's a picture of me. Here's a picture of my wife. If you put us together into some online form, it predicted this is what our kid should look like. This is some sort of intermediate between me and my wife. In contrast, my actual son looks like this. Here's a picture of him. Close to, but not exactly identical to this exact average. Now, oh yeah, he's not usually quite that tan, but he was out in the sun a little bit much that summer. Now, if we look at lots of individuals, we can assess how well the average of the parents' traits predicts the average of the offspring. And then we can use that. We can look at the strength of the correlation, or in this case, the slope of the line, to estimate heritability. So let's imagine a hypothetical thing. Let's take an average height of two parents, and let's say the average height of two parents was 6'0". So let's take another average height of parents to say their average height was 5'0". If the kids, on average, have exactly the same height as the parents, the average of the kids matches the average of the parents, that slope is 1, and therefore the heritability is 1. That is a strong genetic component. In contrast, let's imagine that you see something looking more like this. So you look at the correlation between these two, between the height of the parents and the height of the kids, and you see that in general there's no particular prediction whatsoever. The slope of this line is 0. Therefore, there is no prediction from the average of the parents to the average height of the kids. This would suggest there's no genetic. Well, we can use this to assess heritability. So I have here 4 figures. In figure A here, this is a case where heritability would be approximately 0, where you have short versus tall, the average for the parents, short versus tall, the average for the offspring, and you see the slope there is basically 0. B, you see some relationship. This would be a case where the heritability is about 0.5. C, here you have a very strong relationship there. The heritability is very, very close to 1. In this case, the height of the parents, the average of the 2 parents, very well predicts the average height of the offspring. And here's some real data over here. This is looking at students and their parents. This is from the evolutionary analysis textbook. You see the mid-parent height is very close to the mid-offspring height. The slope there is 0.84, suggesting a very strong genetic component to human height in this case. Now that's interesting, but I have to remind you why this matters. Again, there's medical relevance to this. Let's say your parents get gallstones. Is it worth it for you to alter your diet? Right? Or is this predominantly genetic? Do you as a doctor want to tell somebody, no, give up eating meat completely, when in fact, giving up eating meat may have no effect whatsoever on their likelihood of having some sort of disease? No, you need to be able to identify something that's having a strong genetic component versus a very weak genetic component and more of an environmental effect, so you can practice medicine properly. Similarly, let's say you want to breed a friendlier guinea pig. How much will selective breeding matter? Or is friendliness just how much you handle the guinea pig? Something along those lines. Unfortunately, things aren't always as straightforward as I said. Now, everything we've been talking about ignores the fact that parents and offspring may share some environmental factors as well as genetic factors. There's probably a correlation in food availability and other characteristics between parents and offspring. And what this does is this biases upward the estimate of heritability, because we're assuming anything that's shared in this way is from genetics, but in fact there may be some environmental effect that's shared, especially in the sort of parent-offspring correlation as I talked about. There are other assumptions as well. First, that we're assuming the environment is constant, such as, for example, in that F1, F2 cross we looked at. We assumed that VE was exactly the same in the F1 as in the F2. But in fact, the environment is probably not constant. Estimates of heritability may also be very different if you go to different places because VE may be very different. This is also true at different times. And finally, the amount of genetic variation is also not constant in different families or populations. So let's say, for example, that you study in Australia the heritability of human height. That result may not apply if you went to India or if you went to Japan, things like that. Because the amount of genetic variation is going to be different. The amount of environmental variation may also be different. But despite all this, it's not perfect, but it's a starting point. And it's still useful under particular circumstances. So next class, we'll actually go into another means for estimating heritability, and that is what's referred to as the breeder's equation. We'll also talk a fair bit about population growth. That'll be a little bit of an aside. Thank you, and I hope you'll join us.